Imagine scientists, right, but instead of lasers, they're using super-powered bubbles in a lab to fight bacteria.
Sounds like sci-fi, almost.
Exactly. But today we're actually diving into a real study that does pretty much that. And here's the twist. We'll connect it to the Cütie Catcherz AR comic series.
Ah, yes, Cütie Catcherz. Pronounced cooties, remember, like the playground thing.
Right, cooties. And how its story, which is really fun, also gives us this insightful take on, believe it or not, skin health.
Exactly. The paper we're looking at is called Disinfection of Gram-negative and Gram-positive Bacteria using DYNAJETS hydrodynamic cavitating jets. It's from Ultrasonics Sonochemistry.
Okay, DYNAJETS.
Got it. And the researchers are Gregory Lorraine, Georgia Shaheen, Chelton Sao, Jin-Kun Choi, and Patrick A. Lee from Dynaflow Inc. They're exploring this really novel way to clean liquids using collapsing bubbles, tiny, tiny bubbles.
A super-powered bubble machine. I'm sticking with that. So the goal was basically, can these special jets, these DYNAJETS, effectively knock out bacteria in liquids?
That's the core question, yeah.
Like creating these mini, super forceful bubble implosions to clean water on a microscopic scale. What kind of bacteria were they targeting?
Well, they looked at both main types, gram-negative and gram-positive. Think of it like bacteria wearing different kinds of armor.
Different armor. Oh, yeah.
So for gram-negative, they had things like E. coli, Klebsiella pneumonia, Pseudomonas syringae, Pseudomonas aeruginosa, common names you might recognize.
Yeah, definitely heard of E. coli.
And on the gram-positive side, the tougher armor guys, they used Bacillus subtilis. And get this, they even tested E. coli in actual sewage water.
Sewage water. Okay, that's really putting it through its paces. So how did they actually set this up? How did they unleash the bubble warriors?
Yeah, they use something called a batch reactor system. Imagine a closed loop, right? The contaminated water gets pumped around and forced through these specially designed DYNAJETS nozzles.
Ah, the nozzles are the key part.
Totally. They make the magic happen.
Yeah.
They cause what's called hydrodynamic cavitation. When the liquid speeds through a narrow spot, the pressure drops, forming vapor bubbles. But then, almost instantly, those bubbles collapse violently. Boom! That collapse creates shock waves. That's the cavitation.
Tiny implosions doing the work. Fascinating.
And the researchers measured how the bacteria numbers dropped over time, how many passes or cycles it took. They also tweaked things like the nozzle shape, the pump pressure, how much bacteria they started with.
All right, hydrodynamic cavitation. Little shock waves.
Yeah.
So what happened? Did the bacteria survive this bubble onslaught?
Well, the results were pretty clear. These hydrodynamic cavitating jets, very effective. They significantly reduced the concentration of all the bacteria tested.
So the super-powered bubble machine worked.
It did, but importantly, not equally well against everything. They found some really interesting differences.
Differences. Okay, tell me more. What did they see?
Generally, the gram-negative bacteria, your E. coli, your Pseudomonas, they were killed off faster than the gram-positive Bacillus subtilis.
Ah, the armor analogy again?
Exactly. The researchers even use that, like gram-negative bacteria have lighter armor, making them easier targets for the bubble collapse force.
That makes intuitive sense. But it wasn't uniform even within the gram-negative group, was it? Some were tougher than others.
That's right. For instance, Pseudomonas syringae was knocked down much more effectively than Pseudomonas aeruginosa, even under the same conditions.
Interesting variations.
And E. coli was quite susceptible. It showed significant reduction even when they used lower pump pressures.
Lower pressure worked well for E. coli.
Yeah, and they actually found that using a lower pressure, around 2.1 bar, was more energy efficient overall compared to higher pressures.
So sometimes less is more, at least for energy savings with these bubbles. What about if they started with like a huge bacterial load? Did starting concentration matter?
It did seem to. When they had really high initial concentrations, the killing process was a bit slower at the very beginning. It took a little while to really get going.
Any idea why?
Their thought was maybe the sheer density of bacteria made the liquid thicker, more viscous, slightly dampening the cavitation effect initially.
Like trying to make bubbles in syrup versus water. Got it. And our gram-positive friend, Bacillus subtilis, the one with the heavy armor.
Ah, yes, B. subtilis. It was much tougher. It needed way more cycles, many more passes through the jets before they saw a significant kill rate.
So it wasn't immune, but it put up a real fight.
Exactly. The hypothesis there is that the repeated shock waves gradually weakened its thicker cell wall. It's like chipping away at a strong shield over time, rather than breaking it in one go.
Persistent effort needed for the tough ones. And the sewage test. How did the bubbles handle that messy environment?
Surprisingly well. The disinfection rate for E. coli in sewage was pretty similar to what they saw in the clean lab media.
Really? So the other stuff in the sewage didn't interfere much?
Apparently not with the cavitation process itself.
Rare.
And a big point, they confirmed their DYNAJETS used significantly less energy than traditional ultrasonic methods for disinfection.
That's huge for real-world applications like water treatment. Okay, we know what happened. Let's dig into the why. How do these bubbles actually kill the bacteria? What's the mechanism?
The researchers discussed two main possibilities. The leading theory focuses on mechanical forces.
Physical damage.
Right. The violent bubble collapse generates intense local effects, shock waves, high shear forces tearing things apart, sudden spikes in pressure and temperature. It's a brutal microenvironment.
Sounds like it.
And this physical stress can literally rupture the bacterial cell walls, disrupt their insides, cause osmotic shock, basically destroy them.
And the difference between gram-negative and gram-positive supports this?
Strongly. The fact that the lighter armored gram-negative bacteria were easier to kill fits perfectly with the idea that physical rupture of the cell wall is the main killing mechanism.
Okay, physical demolition. What was the other theory?
The other involves oxidizing radicals. When the water vapor inside the bubbles gets compressed and heated during collapse, it can split into reactive molecules, like hydroxyl radicals.
Like tiny chemical weapons.
Sort of. These radicals are known antimicrobials. But for bacteria in this setup, the researchers felt the overwhelming physical forces were likely doing most of the heavy lifting, more so than the chemical radicals.
So mechanical destruction is the primary weapon here. Now, this study wasn't done in a vacuum. What previous work did it build on?
Right. There's existing research on using ultrasound ultrasonic cavitation for disinfection. It works, but often uses more energy.
Okay.
There were also prior studies suggesting hydrodynamic cavitation, the type used here, was more energy efficient. Some had even used it to break open E. coli to get proteins out, but not always focused on killing efficiency.
I see.
And interestingly, other methods like high-pressure homogenization, which also involves rapid pressure changes, had already shown that gram-positive bacteria are generally tougher than gram-negative ones.
And they link that to the cell wall, too.
Yes, specifically to the thickness of the peptidoglycan layer in the cell wall. That seems to be the key structural difference conferring resistance.
That peptidoglycan layer, the armor thickness, keeps coming up. So what was really new or significant about this specific DYNAJETS study?
It brought a few key things. One, it tested these DYNAJETS against a broader range of gram-negative bacteria than typically studied before with this tech.
Okay.
Two, and this is important, it provided really solid experimental evidence based on that gram-positive versus gram-negative difference that physical cell wall rupture is likely the main kill mechanism in hydrodynamic cavitation.
Reinforcing that mechanical theory.
Exactly. Three, they specifically optimized the process for the more resistant Bacillus subtilis. Four, they showed it worked in a complex real-world matrix, the sewage. And five, they reinforced the energy efficiency advantage of their DYNAJETS technology over ultrasound.
Got it. Significant contributions then. Let's zoom out a bit. What broader lessons can we take from this for general health literacy? Maybe even thinking about skin health.
Well, even though it's about water disinfection, it highlights some universal principles. First, bacteria aren't all the same. Some are just inherently tougher, more resistant to physical stress.
Like the gram-positive ones.
Precisely. And this is really relevant when you consider Cutibacterium acnes. The main bacterium linked to acne.
Yeah.
C. acnes is gram-positive.
Ah, okay. There's the link. So the fact that the gram-positive bacteria in the study were harder to kill, that might relate to why C. acnes can be so stubborn on the skin.
It certainly suggests a parallel, doesn't it? The study also implies that just using brute physical force might not be the best tactic for these tougher bugs.
Like scrubbing really hard might not be the answer.
Potentially, yeah. Just like the bacillus needed repeated cycles, maybe persistent skin issues need consistent targeted approaches rather than just harsh one-off attacks. And the sewage test reminds us our skin is also a complex environment, a microbiome. Treatments need to work with that environment.
Okay, this is where we make that leap I mentioned. We've got super bubbles zapping bacteria in a lab. How on earth does this connect to the Cütie Catcherz AR comic series? The cooties.
It connects surprisingly well through metaphor. In the comic, the cooties, pronounced cooties, are basically stand-ins for Cutibacterium acnes.
The gram-positive acne bacteria we just talked about.
Exactly. Now remember, the study found gram-positive bacteria like bacillus were more resistant to the physical bubble forces.
Right, tougher armor.
Well, in Cütie Catcherz, the cooties, especially the evolved ones, or those hiding in Biofilm Fortresses, aren't easily beaten by just punching them with the cutie pupper's gloves or blasting them with EMP bombs. Nimbus, the hero, learns this the hard way.
So the comic is showing metaphorically what the science showed literally, that brute force isn't always effective against these tougher types.
Pretty much. The resilient cooties mirror the resilient gram-positive bacteria. Just punching harder doesn't work, or could even make things worse in the comic.
Just like maybe overly aggressive physical treatments on skin aren't always best.
Could be. And the study needed more effort, more cycles for the gram-positive bugs. In Cütie Catcherz, what do they introduce? Specialized tools.
Like the phage darts.
Phage darts, probiotic pods for microbial balance, buster gel, whips of wrath for biofilms. These targeted tools in the comic parallel the need for optimized, maybe gentler but more persistent or specific strategies required for the tougher bacteria in the lab.
And Nimbus has mentors, the Paw Patrol, right? They teach him about balance and understanding, not just attacking.
Yes, and that resonates directly with the study's implications. Optimizing pressure, needing multiple cycles. It's about understanding the target and the environment. The Paw Patrol teaching restraint and understanding the biome mirrors the scientific need for balance and targeted approaches in complex biological systems, like our skin. It's not just about annihilation.
What about the comic showing cooties evolving, getting tougher, building those biofilm fortresses? Does science echo that?
Absolutely. That directly mirrors real-world challenges like antibiotic resistance in bacteria, or the formation of protective biofilms by C. acnes, which makes acne harder to treat.
Biofilms are like actual bacterial fortresses.
They really are. And just like the study found high bacterial loads slowed things down initially, the comic often shows severe cootie breakouts being harder to control at first, needing persistent effort. It's another neat parallel.
So Cütie Catcherz isn't just a cool story, it's weaving in some real scientific concepts through its metaphors. What are the final takeaways for you, the listener, connecting Cütie Catcherz, this study, and skin health?
Okay, first, the cooties are tough in the comic, partly because their real-life counterpart, C. acnes, is gram-positive, and science shows these types are more resistant to physical force. So the comic showing that just popping isn't the answer. It actually aligns with the science about their stronger cell walls.
Right, brute force isn't the solution in the story, or potentially for your skin.
Second, smart, targeted approaches win. Nimbus learns this, needing specific tools for specific cootie problems like biofilms. This mirrors the need for maybe specific skincare ingredients or routines tailored to the problem, rather than just harsh, broad attacks that might disrupt the whole skin biome.
Like the study optimizing conditions for different bacteria. Precision matters.
Exactly. And third, patience, precision, and balance are key. The Paw Patrol emphasizes this, consistent care. The study showed resilient bacteria needed optimized pressure, multiple passes. It's about fostering a healthy, balanced skin environment, understanding that different issues, like different cooties or different bacteria, might need different consistent tactics.
It really emphasizes balancing the biome over just trying to wipe everything out.
That's a huge takeaway. So, yeah, this deep dive uncovered this really cool parallel, didn't it? Between hard science on bubble disinfection and the imaginative storytelling in Cütie Catcherz, both shedding light on bacterial resilience, especially gram-positive types, and what that means for understanding skin health, particularly acne.
It's genuinely empowering, I think, seeing how science can be reflected even in places like an AR comic. Hopefully this gives you, our listener, especially if you have darker skin and face unique acne challenges, a fresh perspective on your skin health and the tiny world living on it.
And it makes you wonder, right? What other everyday things have this hidden layer of science, these surprising connections or metaphors just waiting for us to look a little closer, maybe something to ponder?
You got the cooties, we got the cure, fighting breakouts with facts and a whole lot of lore. It's Cütie Catcherz, beneath the skin, where science and story are about to begin. Let's go!
That's a wrap on this episode of Cütie Catcherz Beneath the Surface, where science meets art and technology, and skincare gets a superpower treatment. If you learned something, laughed a little bit, or just loved the blend of art, culture, and biology, make sure to follow the journey at cutiecatchers.com and stay up to date on Instagram at Cütie Catcherz. Catch every new episode on Spotify and YouTube. Just search Cütie Catcherz Beneath the Surface. And remember, the cooties are real, but so is your power to fight back with knowledge. This is produced by me, Stephen Christian, of Stuck on an Island, in collaboration with Iltopia.